Hello friends, welcome to Dr. Ossai Physiology Academy, DOPA for short. This is the place where we make the learning of physiology easy, exciting, and effective. Thank you for joining me. So now we're going to be doing lecture 4, Tubular Reabsorption and Secretion. Remember that we talked about there are 3 basic renal processes: glomerular filtration, then tubular reabsorption, and secretion. Glomerular filtration takes place here, and we've sufficiently dealt with that, how it relates to renal blood flow, glomerular filtration rate. But something you need to always remember, that learning how the body functions, the step-by-step processes, which is what physiology is all about, can teach you vital health lessons, life lessons, and even management lessons. Yeah, so let's get started. So we've finished with glomerular filtration, and we said something that glomerular filtration rate, GFR, is equal to 125 mils per minute. Okay? What does that imply? It means that in one day, you get 180 liters of filtrate per day. This is a whole lot. The kidneys filter 180 liters of fluid in one day. But the urine that you pass out in a whole day is usually no more than 1 to 2 liters. What does that tell you? That a lot of fluid has to go back into the blood. So we're going to be dealing with that. What are the mechanisms by which the renal tubules reabsorb substances, and also there's a process of secretion? So when we talk about reabsorption, it shouldn't be abstract to you. It's basic transport mechanisms which you are supposed to have learned in general physiology. You know, when you learn about everything about physiology, when you want to get a good grasp of it, you go to the cellular level. So when you're just talking about reabsorption, it is cells that are dealing with those things. So first of all, transport mechanisms, just a recap, you have active transport and passive transport. Okay? So these 2 transport mechanisms, they play a role in the movement of substances. And these things, they are across membranes. So when I'm talking about reabsorption, it's talking about transport across membranes. That's why when we were talking about the general structure, we mentioned that there are cells here. All these things, this nephron, this tubule in the nephron, they are lined by cells, a single layer of cells, so that movement of substance will be very easy. So it's very thin. Okay? So there are cells, cuboidal cells, flattened cells in this thin area, and so on. So in this active transport, you now have the primary active transport and the secondary active transport. The passive transport, you have the simple diffusion and the facilitated diffusion. So these processes, they all play a role in helping the movement of solutes, electrolytes, and other organic substances that are filtered. Because when filtration takes place, it only excludes blood cells and very large plasma protein. Every other thing, glucose, amino acid, nutrients, everything enters. But the body needs most of those things back. It needs them back. So we're going to be dealing with it systematically. Remember, we have different segments: proximal tubule, You have the loop of Henle, distal tubule, and collecting duct. So we're going to be taking it segment by segment. All right. So now this proximal tubule or proximal convoluted tubule, what happens there? The proximal convoluted tubule, the important fact you must always remember is that it is the main reabsorber, is the mass transporter of this tubule. A whole lot of reabsorption happens here. Up to 60% of the reabsorption of water, this 180 liters you are seeing here in a day, 60% of it is reabsorbed here. All the solutes that are important and needs to go back to the body, also glucose, sodium, potassium, chloride, bicarbonate, and so many like that. I need to go back. About 60% happens here. Note that. Okay? So we're going to start with each and every one of these important. In other segments, we'll just focus mainly on water and sodium. They are the main, water is the main thing there. Okay? And sodium, the main solute. Because many of all these solutes, in a way, they are coupled with the transport of sodium. All right? So let's start with sodium in this proximal tubule. How is sodium transported? Now, like I said, all these places here, they are cells. A thin layer of cell, okay? Then this place here is the interstitium. Interstitium. When you talk of interstitium, you have interstitial fluid here. Then, from this efferent arteriole, you now have what? Peritubular capillaries. So, what happens in the process of reabsorption? Fluid enters inside this tubule from here. Then, they have to pass through this cell. So, this cell, the part of the cell that is facing inside the tubule, this is a cell, the part of it facing inside is called the apical membrane. Okay? Or luminal membrane, lumen. Luminal membrane. All right? Then this part that is now facing this interstitium is called basolateral membrane. So when substances needs to be reabsorbed, it comes from the lumen, it will pass through the apical membrane, enter into the cell, then pass from inside the cell through the basolateral membrane and enter into the interstitium, which has interstitial fluid. Then from the interstitial fluid, they now gain entrance into the peritubular capillaries, which enters the circulation, the bloodstream. All right? So now we want to talk about how these substances, they move. Sodium. We want to start with sodium. Remember that every cell in your body, every single cell, they have a certain carrier protein that uses energy. Is the main active transporter of every cell. What is that transporter? It's called sodium potassium ATPase. All right? Sodium potassium ATPase. And what does it do? It is fond of transporting 3 molecules of sodium outside of the cell, and at the same time transporting 2 molecules of potassium inside the cell, takes it outside continuously. It does not stop. Always taking sodium outside the cell, bringing potassium. So it ensures that at every point in time, the concentration of sodium within every cell is always low. Okay? And the concentration of potassium is always high inside the cell just because of the action of this particular transporter. It's an enzyme, ATPase, okay? So that's what it does. So what is the implication? What's the consequence of that? How does it relate to what we're talking about, reabsorption? Now, these are cells lining this proximal tubule, okay? So it means that this transporter is doing its work. So it's taking potassium out from the basolateral membrane into this interstitial fluid continuously. So inside has low. So the filtrate that enters has more concentration of sodium. It means that there's a concentration gradient that favors the diffusion, simple diffusion, okay, which is passive transport of sodium from within this lumen into the cell. The concentration is low inside the cell, lower than what has been filtered. So there is a passive transport. The energy there is that gradient that has been created because of the action of this. So because of that energy with which sodium enters into the cell as a result of that large concentration gradient, it uses it to carry many other substances. Okay? So that is called what? Secondary, look at it here, secondary active transport for most of these substances, depending on the gradient. Now we're going to talk about it because sodium, one of the major solutes that it carries is glucose. And glucose has something that is opposite, a property that is opposite that of sodium, in the sense that glucose is always higher inside the cells than outside. So whenever glucose wants to go inside the cell, that means it's going from where it is less to where it is greater. It is acting against the concentration gradient and it needs energy. So that's why it's called secondary active transport. Okay? And amino acids and all of that, they are always higher inside the cell. So, but sodium is going along its concentration gradient because it's lower inside. So as it's going, it's carrying other substances. So the energy is from this primary one. That's why it's called secondary active transport. So that's what happens in this place. So because we've mentioned glucose, let's just talk about it. We've talked about sodium now, how it is transported. Is based on that gradient created by this. So it carries glucose. Now, how does glucose pass through with sodium and now enter into this other interstitial fluid and enter into the blood? They always use transporters. Now, the transporter that carries sodium and glucose from this lumen into the cell, remember, from the apical membrane, carries it into the cell. It is called sodium glucose transporter. Okay? You have 1 and 2. I think the one there in this place is 2. All right? Sodium glucose transporter, SGLT2. That's what, so this thing is secondary active transport because glucose is going against concentration gradient. Then when glucose now enters into the cell, it's not staying there. It needs to enter the bloodstream. That's where it's supposed to be because it came from the bloodstream, filtered down. So it needs to enter back into the bloodstream. So what happens is that glucose is higher now inside the cell, and it wants to enter the interstitial fluid where it is lower. So that means it's no longer active transport, it is now passive transport. And the transporter that takes glucose across the basolateral membrane into the interstitial is called glucose transporter, GLUT2. Okay, you have GLUT1. All right, so that's what happens. And this is called facilitated diffusion. All right, so there's something, a concept that is very important that we need to mention as it relates to glucose transport. Now, in all facilitated diffusion processes, okay, they exhibit saturation kinetics, in the sense that you have a cell, let's say this is the cell, and the carrier protein is located at the membrane all around. Okay? The truth of the matter is that these carrier proteins are limited. They have a specific number of glucose, in this case glucose, that they can bind. So what happens if the molecules of glucose now occupy every one of them? Let's say there are about 1 million, and 1 million molecules occupy everything. What do you think will happen? That means this particular cell will now be incapable of transporting glucose from inside the cell to the interstitial fluid. That's what happens. So in the case of this tubule, if the concentration of glucose, which normally doesn't happen, but if it happens that the concentration of glucose that is being filtered from the blood into it is very high, that it occupies all of this, the excess glucose will have to be excreted in the urine. That's the implication. Okay? And you will see that in people that have diabetes mellitus, DM. They usually excrete glucose, sugar. That's why you can see some ants, they gather their urine because their urine is sweet, contains glucose, sugar. It's because of this mechanism. Okay? The carrier protein is occupied. It can no longer take glucose inside, so glucose is excreted. So keep that in your mind. And it happens at a level, the blood glucose level of about 200 milligram per mil. Okay? So this, at this level, it is called what? It is called transport maximum of glucose. Transport maximum. Okay? So we have dealt with sodium, glucose. So let's talk about water. How is water transported? Water is very easy. It's normal through osmosis, okay? So, but it does that through what they call water channels. Water channels. And they are known as aquaporins. We have aquaporin 1, aquaporin 2, depending on the part of the renal tubule. Okay? So aquaporin, they are known as water channels. So the water from this place through osmosis enters into the interstitial fluid and enters into the bloodstream. A lot of, about 60% of water is reabsorbed. And that's because, remember that this proximal tubule has microvilli, okay, brush border. That's why it reabsorbs so much of both water and solutes. About 60% of water, 60% of solutes. So it doesn't dilute or concentrate the filtrate. The filtrate that leaves the proximal tubule is still the same osmolarity of about 300 milliosmoles per liter, because equal number of water, equal number of solutes get transported. So what other substance is reabsorbed? Chloride. Okay, when water, sodium, and all of that, they are reabsorbed. So the concentration of chloride slowly increases as it's going down. So it now favors the diffusion of chloride. It's called anionic diffusion. Okay, so what else? Bicarbonate. Bicarbonate is also something that is very important and needs to go back into the blood. How is it reabsorbed? Very specially. Now, bicarbonate cannot cross the cell directly, but it is needed. So how does it do? So the body devised a means of transporting bicarbonate in a special way, indirect way. So this is what happens. So this bicarbonate, how does it cross? What happens is that it combines with hydrogen plus hydrogen. So what do you think it will give you? To give you carbonic acid, H2CO3. And this is very unstable. It's carbonic acid. It easily dissociates into what? Carbon dioxide and water. Right? So carbon dioxide easily, very quickly, can easily cross the membrane, cross and enter into the cell. So when the carbon dioxide that has crossed enters into the cell, it now goes and recombines with water inside the cell. So the reverse takes place. It recombines with water that is inside the cell, and it forms carbonic acid, which also now dissociates into bicarbonate and hydrogen. So that's how bicarbonate has now reformed. So bicarbonate cannot cross directly. It will have to go through indirectly by breaking up into carbon dioxide and water. Carbon dioxide crosses, then recombines with water inside and dissociates again. And that's how you get bicarbonate. But we'll discuss that in acid-base, because this is a base. It's important in acid-base balance. We'll talk more about that later in later lectures. Okay, so we talked about hydrogen combining with bicarbonate. Where did the hydrogen come from? So hydrogen is another important solute that is transported, but its own is in the area of secretion. You know, when it's going inside, reabsorption coming in. So it combines with sodium. No, sodium is going inside. Sodium is being reabsorbed. So, but as sodium is being reabsorbed, there's a particular transporter that is called sodium hydrogen exchanger. So it's taking sodium in and taking hydrogen out into the lumen. Do you understand? Taking sodium from this lumen into the cell and taking hydrogen from inside the cell into the lumen. Opposite direction. It's an antiporter. Call it antiporter or counter transport. Okay? Sodium hydrogen exchanger. So what other transport mechanism do you have in this proximal tubule that helps? Another one is potassium hydrogen transporter. So you have potassium hydrogen potassium hydrogen transporter. It takes potassium in. Now remember that potassium naturally is high inside the cell, okay, and low in the surrounding. It's going against its concentration gradient. High inside, but it's taking it inside and taking hydrogen into the lumen. So it's a transfer, it's exchanges. So because of that energy it uses to take potassium inside, you can also call it ATPase. Okay, needs a lot of energy. Potassium, sodium, ATPase. So about 90% of potassium is reabsorbed because of that transporter. Although later it will now be lost again through secretion at the distal part. But at this proximal part, 90% of potassium is lost. Then finally, let's talk about another one. Calcium. Calcium is a very important solute that needs to be reabsorbed. So about 90% of calcium also is reabsorbed in this proximal tubule through an active transporter known as calcium ATPase. Okay, so that's quite straightforward. Then urea also is reabsorbed at this place. About 50% of urea is also reabsorbed in this proximal tubule. So, but it goes along with water. Once water is reabsorbed, the concentration of urea rises, so it now favors urea going. So it's called solvent drag. Water drags it along. So that is the proximal tubule. You see a lot of things happen in this proximal tubule. Unlike the other places, we'll just be focusing on water, whether water is reabsorbed or not, solutes, majorly sodium, and so on, okay? So we've talked about the proximal tubule. The secretion is not much in the proximal tubule. One of them is ammonia. Okay? Ammonia is secreted in the proximal tubule. So, but it exchanges with sodium. A lot of things use sodium for their transport. Then hydrogen, like we've mentioned, is secreted in this proximal tubule. So now we're going to move over to the loop of Henle. It's another special place. So we'll do that after this break. Right, so welcome back. So we've dealt with this proximal tubule where most of the show happens, okay? A lot of the reabsorption happens. So now we're going to start with the loop of Henle. So let's clean all this. Okay, so now the loop of Henle, like has the thin segment, thin descending, thin ascending, then thick ascending, okay? This thick ascending limb. So what happens is that this loop of Henle, by the virtue of the fact that it's thin, not much happens there. But there's something that is significant about this loop of Henle. After the proximal tubule, where we talk about reabsorption of so many substances, now we are just going to focus on majorly water and sodium are the two major solute solvents, okay? So what happens with water in this loop of Henle? About 20% in the whole loop of Henle of water is reabsorbed through the normal process we talked about. But that 20%, all of it is reabsorbed in just this descending limb. The whole of the ascending limb, both the thin ascending and thick ascending, is impermeable to water. Okay? So this part takes in water. It's moderately permeable to solutes, but majorly water is freely transported in this thin descending limb of loop of Henle. Then this whole part is impermeable to water. But what? It is permeable to solutes. So what do you think it will result in? Water is not leaving and entering the interstitium, but solutes are leaving from inside here and entering. Sodium is leaving and entering. What does that mean? That means this segment of the fluid becomes more and more dilute. So that's why it is called the diluting segment. It's called the diluting segment because it does not allow water to move, but allows solutes to move. So solutes are lost from the tubule at this point. So what's the transport mechanism of sodium that transports sodium from inside this tubule into the interstitium? It is called 1 sodium, 1 potassium, and 2 chloride. Very funny transporter. It transports 1 molecule of sodium, 1 molecule of potassium, and 2 molecules of chloride. So that's the name of the transporter that transports sodium. We are focusing on sodium. Transports sodium and chloride inside. Okay? So we are done with the loop of Henle. What is the secretion that happens there? Mainly the secretion is the hydrogen, that exchanger, hydrogen. As sodium is going, there's also an exchanger that exchanges hydrogen into. So it's secreted into the tubule. But remember that this hydrogen, if just like a rule of thumb, hydrogen is secreted in every part. Proximal, loop of Henle, distal tubercle is secreted into the tubule, except the thin limb, just this thin part. So hydrogen is secreted in the ascending limb of the loop of Henle only. The thin descending, the thin ascending, hydrogen does not do anything. It's not secreted. So this is the only part where hydrogen is not secreted into the tubule. So we are done. That's for the loop of Henle. Now, remember when we're talking about the histology, we said that this thin ascending limb has similarities with the early part. From here is the distal tubule. Okay? Distal. It has a lot of similarities with the distal tubule. So what happens there? The same thing that happens in this thick ascending limb happens here, in the sense that it is impermeable to water. So it's still continuation of the diluting segment. It gets more dilute because solutes are entering, but water is not moving. Water is not entering because there are no water channels. But the only difference in this early part, when I'm talking about distal tubule, we usually talk about early distal tubule, late distal tubule. Early distal tubule has similarities with thick ascending limb. Late distal tubule has similarities with the collecting duct and all of that, okay? So the early distal tubule, the only difference it has with the thick ascending limb is the sense that what transports sodium is different. It's just sodium chloride transporter. Instead of 1 sodium, 1 potassium, 2 chloride transporter, it's just sodium chloride transporter. Okay? That's what transports sodium into this. All right? So now we are now in the late distal tubule and collecting ducts. Now here's the cortex, here to here. Here is the outer medulla, inner medulla. So a lot has happened up to this point. A lot of reabsorption and secretion. So the good thing I need to know about this late distal tubule collecting duct is that is where regulation takes place. the final amount, what will determine the final amount of what is excreted, solute that is excreted, and water that is excreted. When I'm talking about solute, majorly sodium, okay? So the amount of sodium that is going to be excreted will be determined by what happens in this place. That means it varies. It's not the same, okay? All these early parts, they have specific percentages that are constant, whether you like it or not. This particular amount of sodium and water will be reabsorbed and transported up to this part. But from this late distal tubule collecting tubule, it varies depending on the needs of the body. So what do we mean by that? Now, in this late distal tubule, remember we have two different types of cells there. The principal cells and the intercalated cells. Principal cells and intercalated cells. So what happens is that these principal cells and intercalated cells, they perform opposite functions in terms of potassium transport, okay? And what do we mean by that? Just use this code, PSIR. It means that the principal cells, they secrete potassium. The intercalated cell, they reabsorb potassium. Can you see the difference? The same potassium, the same different, these two cells, they handle it differently in the same distal tubule and collecting duct. While the principal cells, they secrete potassium, the intercalated cells, they reabsorb potassium. But both of them secrete hydrogen. Like we said, hydrogen is always secreted in all parts except the thin part of the loop of Henle. Okay, so now what happens with water in the late distal tubule and collecting duct, and what happens to sodium in those segments? Remember, that's where they are regulated. Now, water will be reabsorbed depending on the concentration of a hormone called antidiuretic hormone. Let me write it in full. Antidiuretic hormone, known as ADH for short. It has another name called vasopressin because at some concentrations, it helps to constrict blood vessels. Vasopressin, okay? So normally, that those late distal parts, late distal tubule and collecting ducts, they are not permeable to water, just like the early distal tubule and the thick ascending limb. But if you have enough of this hormone secreted, this hormone now helps to stimulate the synthesis of those aquaporins. They are proteins, water channels. So they are now, those water channels are now inserted, and then water can now be reabsorbed. But if you don't have enough of ADH, there are no water channels, it is impermeable to water. So that's the regulation. The regulation is done by hormones. So for sodium is also regulated, okay? There is a hormone called aldosterone. Aldosterone regulation. ADH regulates water transport. Aldosterone regulates what? Sodium transport at the late distal tubule and the collecting ducts. So if you have a lot of aldosterone, more sodium will be reabsorbed. If you have little aldosterone, little sodium will be reabsorbed. So that's what changes the game. How much of sodium is finally excreted in the urine is determined by aldosterone. How much of water that is excreted in urine is determined by ADH. And they are secreted, they are released, those hormones are released based on the concentration, the osmolarity of the blood, the ECF osmolarity. That's what determines how much they are released. Okay, we'll talk more about that later. So, and we said the transporter of water is aquaporins. The transporter of sodium is different. Look at all these places. Sodium is always changing its transporter. Very funny. So look like that. So this aldosterone at the distal part, the channel is called ENaC, epithelial sodium channel. Okay, so that's what it uses to now transport sodium under the influence of aldosterone. So take note of it. ADH influences the creation and synthesis of aquaporin that helps to transport water. Aldosterone does similar thing, but for sodium. Synthesizes this protein and that helps to transport sodium reabsorption. All right, secretion, we have already mentioned that hydrogen is constantly secreted. Then, depending on whether it's principal cell or intercalated cell, principal cells secrete potassium. Intercalated cell reabsorb it. So that's for the secretion and reabsorption. So now we are done with tubular reabsorption and secretion. What do you think we can learn? What lessons do you think we can learn from there? A very vital lesson is that if you look at all this, the journey, all this place, there's constant reabsorption and secretion. The volumes, the percentages are consistent. Where it varies is at the distal part. So the same thing with the way you organize your activities, your life. There should be a part of your life that is stable, unchanging. That is unchanging, stability. And those things have to do with the principles and the values I live by. They don't change, no matter what the circumstances. But there should be other parts of your life that are flexible, like the late distal and the collecting duct and all of that are flexible, can regulate and you can adjust. Okay? So those things have to do with your methods, your techniques and all of that. You need to be flexible. You need to easily adjust those things to fit the circumstance. That's how to succeed. There's stability, there's flexibility. All right? So the next video, we are going to talk about concentration of urine. All these things, this is normal reabsorption and all of that secretion. But we need to talk about the fact that sometimes you need to excrete dilute urine, other times you need to excrete concentrate. How does the body do that? How does the body know which one to do? That will be in the next video. But remember that you can check out some materials that we have, some resources in renal physiology and in other parts of physiology. We have written some books. Check it in the description box. You'll see the link there. I'll direct you to Okada Books, soft copy, just for a token for further reading, so you get a better grasp of these things. All right? Thank you.